Today we're going to talk about how to basically make a DNA profile. We're going to kind of talk about the old method first. The first way that they created DNA profiles, because they use parts of the old method in the new method, and it's kind of important for you to understand both so everything kind of makes sense. So for DNA profiling, it was a technique used by scientists to distinguish between individuals of the same species only using their DNA samples. And the process was discovered by this guy, Alec Jeffreys, in around 1985. So it hasn't been around for a super long time in comparison with some of the other techniques that are out there. So the basic process of profiling, okay, step one, the cells are broken down to release DNA via extraction. And we've already talked about how to do a DNA extraction, right? We've talked about how they get the DNA out of the cells, the whole process that you go through with the lysis solution and the isopropyl alcohol, etc. So step one is basically to extract the DNA out of the cells. And if there's only a small amount of DNA available, then they have to also amplify it using PCR. So PCR is kind of lumped into this step because if they need to do it, they do it in step one, okay? For step two, the DNA is cut into fragments using restriction enzymes. And we already talked about what restriction enzymes do, right? They use recognition sites to cut DNA at specific locations. So they pick a specific restriction enzyme and they put it into the DNA, and the restriction enzyme goes through and it cuts the DNA into fragments of all different sizes, just random fragments of all different sizes, depending on what your base pairing sequence is. Okay, so that's step one and step two. Then what they do is the fragments are separated on the basis of size using a process that we call gel electrophoresis. And gel electrophoresis is where they take this apparatus right here that you can see, and they put the DNA into it, and they use an electrical charge to separate the DNA from smallest fragments to biggest fragments. So remember how I had you guys, when we talked about restriction enzymes, I had you guys kind of putting them in order according to size. Well, this is the apparatus that actually does that. So let's take a look at how this works a little bit. So this is kind of the top right here, okay, and this is the bottom. So top and bottom. These are the well plates. So these are the little wells that the DNA goes into. There's 1, 2, 3, 4, 5 of them here. Okay, right there are the wells. And so what they do is they load the DNA that they just extracted, they made copies of, and they cut. They load it into these little wells. And then what they do is they put what's called a buffer solution, which is like a clear liquid over top of it. And they hook up this whole apparatus to a battery. And on this side, there's a negative electrode, and on the other side is a positive electrode. Well, DNA has a negative charge. So it doesn't like to be next to the other negative. Remember, positives and negatives attract each other. So what happens is it wants to move from this side down towards the positive side. And as it does that, the smaller fragments are able to go through the gel quicker and further than the bigger fragments are. Okay? And so it ends up separating all the fragments according to their size. So the fragments that make it all the way down here are super small. And the fragments that don't make it very far, like these ones up here, are the very large fragments. Okay? And so that's how the process works. Okay? It basically takes electricity and sends it through this apparatus, and it moves the DNA through the gel, with the shortest ones going the furthest, the longest ones not going quite as far. And then what they do is they add a radioactive material called a probe to the gel, which combines with the DNA fragments and produces a fluorescent image. So basically what it does is it makes the DNA visible, because DNA is clear or invisible. So a probe right here is a sequence of DNA that's complementary to the fragment you're interested in. So if the DNA that I was interested in was AATT, okay, if that was like the small segment that I wanted, the probe would be TTAA, right? So it's like a little marker that goes in and then it attaches itself to that location in the DNA. And it goes in and it bonds to that specific sequence, and then they add a fluorescent marker to it, and then it shows up. So this is what it looks like after they've added the probes to it. It just makes the DNA fragments visible. Okay, so you can see like on this line you had 1, 2, 3, 4, 5, 6 fragments of DNA that the restriction enzyme cut. These are the short ones, these are the long ones. This one you can see matched. Can you see how they were the same? They were in the same locations, in the same spots. So this person in this well and in this well had the same DNA. This person and this person had the same DNA. And this person and this person had the same DNA. So if you were looking, say that you had, right, stuff from your crime scene, and then you had victim or suspect 1, suspect 2, suspect 3 type of thing, you would be able to tell who it matched by taking a look at what the bands did. And then for step 5, it's just the pattern of fragment distribution is analyzed. Basically, you're going to take a look at all the bands and you're going to see if anything matches. The chances of 2 people having exactly the same DNA is 30,000 million to 1 except identical twins, which basically means it's impossible. They have yet to find anybody who has any matching DNA except identical twins. So if they're able to find a match to your DNA, it means that it's for sure yours. So taking a look here, if this was my electrophoresis printout that I got, and this was my suspect's profile, the person I think did it, this was blood from the crime scene, and this was my victim's profile, does your suspect match the blood from the crime scene or does the victim? And what you want to do is you just want to see what the location and the size of the bands are. So you can tell that the suspect matches. These bands are the same, these 2 bands are in the same location, this band is in the same location, these 2 bands are in the same location, and this one. And even though the victim has some of the bands similar, they're not exact, and so they don't match. So the suspect matches the blood from the crime scene 100%. And DNA is an individual characteristic, which means that it's not going to match anybody else except that person if it's that person's DNA. They also use it for paternity testing, and that's deciding whether or not the child belongs to a father. So by comparing the DNA profile of a mom and her child, it's possible to identify the DNA fragments in the child that are absent from the mom and therefore must be inherited from the biological father. So keep in mind, you get half of your DNA from your mom and half of your DNA from your dad, okay? So with that being said, okay, if this is the child's DNA, okay, the mom matches this one and potentially this one, right? The mom does not match this one. The mom doesn't match this one. The mom matches this one, doesn't match this one. So 1, 2, 3 of the 6, those 3 belong to the mom. That's the ones the mom got. So now you have to say, okay, with the ones left over, are they in line with the dad? So this one is, right, same spot. This one is, this one is. So this child is this man's, okay, because those were the ones that matched exactly. So basically when you're doing DNA profiling for paternity tests, you have to eliminate out which ones could potentially belong to the mom first, and then whatever's left over, if that matches the dad, then you know that that's the case. Now, if 2 of these matched, but the third one, say, wasn't in the right location, then you would say no match. They all have to be in the same spots, because for paternity tests, all of the DNA has to be accounted for, because you get half from your mom and half from your dad. And we'll do some more practice with this as we go this week. You guys will do a DNA electrophoresis lab. You will do a paper lab with it. So you'll get a lot of practice at how this works.